There we go. Perfections. All right, good morning, West Hill scientists. I'm Ms. Bosco, I'm the science department head. We are joined today by a professional scientist from Regeneron. Please give a warm welcome to Mr. Emmanuel.
Good morning, guys.
Good morning.
Thank you. My name is Emmanuel, and I'm a senior associate scientist at Regeneron. Thank you for having me here at West Hill. I wanted to do this program for one reason, because it's not such a long time ago when I was in high school and right around 11th or 12th grade. Well, in Nigeria, we call it a different thing. It's called secondary school, and we call it senior secondary 5 and senior secondary 6. So it's like 11th and 12th grade. At the time, for me, someone who, supposedly cares about their future, cares about career, wants to know what they want to do, I was kind of like stuck between a rock and a hard place. And that is because so many things were not clear to me at the time. I was in Nigeria. My parents were here in the US. I was living with my grandma, and she was into farming. So I farmed with her a lot. It started actually when I was in elementary school. She would grow crops like corn, melon, and things like that. She even had some chickens and goats and stuff in the pen at the house. It was a life, but I was a student. And I didn't want to do farming as a career. I mean, who wants to, right? But that was all that was in front of me. The thing that kind of like started to shake me was thinking about what I want to do in the future versus seeing where I am right now. It's like two different things. What I wanted to do in the future, I didn't know. But where I was right now was farming. That was all I knew. So what happened? I let my curiosity push me. When I say my curiosity, I mean the curiosity of what I'm doing, like farming, what is it about, and, all of that stuff. I feel like 11th grade, 12th grade, for students, maybe other people might not understand it. Some people who care about the future start asking themselves questions like, what am I going to do in college? Do I know what I want to do in college? What am I going to major in college? Am I going to do well in college?
They're all asking those same questions.
Yeah, exactly. If you've ever asked that question, can I get a step? Yeah. That's how I know I'm in the right place. These questions are valid, but everybody around you just sees the celebratory stuff, right? Senior trips and prom, graduation. But deep inside, there's always this question about, what do I really want to do? Do I know what I want to do? If I get in college, am I going to succeed? What I'm here to share with you is my own story, how I figured it out, or how it worked out for me. And that's what I call the STEM story. So thanks for having me, okay? Next slide, please. All right, today I'm going to tell you a little bit about myself, who I am, and then I'm going to tell you about my journey to becoming a scientist. How did it work out? Was it planned from the beginning? Did it just happen? What led me there? And then I'm going to tell you what I do at Regeneron, my team and I. What do we do every day when I go to work? And then I'm going to tell you about the instrumentations we use at work to actually do this work that we do. And then we're going to discuss the Science News article. Next slide. All right. So who am I? The people call me a scientist. Yes, I perform science. I don't know if that makes me a scientist, because there's astrophysics, there's like a bunch of different things that you can call science. But yes, I am a scientist because I do science every day. I love to hike. That is me at the Diamond Head Hike Trail in Hawaii. I have a daughter. She's only 10 months old, so she's very beautiful, Serafina. At the church, I'm part of the music team. I'm the music director. I have a little bit of a choir that I work with. I record music sometimes in my free time, at home, in the studio. And then sometimes I perform with my friends. And then I do what we call Go Love Nations, where I go back to Nigeria and I collaborate with the healthcare professionals in Nigeria to provide free healthcare for people, free medications, right? We have people line up and then they come in, they get diagnosed, and then we provide them with medications, either the ones we brought from here or the ones we bought from pharmacies in Nigeria. And then, we kind of follow up with them over a period of time to see how they're doing. Because when I come back to the States, we'll have people there, local healthcare professionals and volunteers who follow up with them to see how they're doing. Thank you very much. So that is something that I work with. I call it the Medical Missions Organization. So that's a little bit about myself. And as a father and a scientist and music director and hiker and all of these other things, life seems rich. And when you see something like this, you might seem like, somehow this adult figured it out. How are you able to just figure it out? And then, you just know what you want to do. Did you know what you want to do from the start? How did it all come together? The truth is, ideas are not fully formed in the beginning. And I'm saying it to say, if you don't know what you want to do right now, it's okay. If you don't know where you want to go right now, it's understandable. But one thing that helped me, like I said in the beginning, was curiosity. If you follow your curiosity, you would never go wrong. What does that mean? The thing that bothers you in the mind all the time, the thing you think about all the time, most likely than not, leads you towards your own path. This is not poetry, it's true. Because that is what puts the light in your eyes. That is what you always look up whenever you go to the internet. That's what you always read up on your own before somebody tells you to. That is something you want to know more about. I didn't want to do agriculture, like I said from the beginning. But because I was in that environment, I started asking questions. How is it that my grandma could have some seeds from years ago, and then eventually, years later, we plant it and it grows? That blew my mind. Somebody else might not care about it. Right? How do we actually, we bury this stuff in the ground and it sprouts out and it grows, starts, one corn gives us a bunch of maize and thousands of seeds. The curiosity started to drive me. I didn't think of making a career out of farming. I didn't think of it that way. I was just asking questions. And then it turned out that in school, around 10th and 11th grade, I had to pick a course between home economics and agricultural science. It's a no-brainer. I picked agricultural science. And then the class started telling me things that helped me understand what was going on in the farm. In class, we'll learn about, how the origin of plants, how plants started from algae, green algae. And then eventually how they invaded the land, right? With the different evolutionary adaptations. Started to learn about gymnosperms and angiosperms and, the way they actually generate their gametes and sporophyte phase and the gametophyte phase. It didn't make sense. But then I'd go home and I'm telling my grandma, this is what we call gametophyte phase. This corn that we planted, there's it. And she doesn't get it. She doesn't care. But to me, it was just blowing my mind, right? And I was looking for someone to go tell it to because I was excited about it. Again, I wasn't thinking about career at that point. I was just answering questions that were right in front of me that I cared about. And then what I didn't realize was that was building my interest in the life sciences. Because plants, eukarya, right? Plants, animals, are part of the eukarya domain. And we share a lot in common. In 2012, eventually, finally, my immigration papers came through and I flew to the US. And that's when I started college. Now, between 12th grade and college was a 6-year period. I had a 6-year gap. Because I was waiting to migrate to the US and I didn't know what to do. So I would be in the farm a little bit, out in the shop a little bit. It's like a lot of confusion for me. And when it came to the US, I had this quiet desperation. It was like a huge fear. My high school was in Nigeria. Maybe it's a low quality standard of education, some might say. Now, I'm gonna become a freshman competing with school, I mean, high school kids fresh out of, 12th grade, and we're gonna be freshmen in college. The brains are sharp. They studied high school here. They know what is going on. I have a 6-year gap from a third world country. How am I gonna compete? The fear was enormous. I was almost sure I was gonna fail. I didn't have an option at all. Agriculture, who cares? Here, this is New York, right? Nobody does agriculture here. It's not a thing. But that was all I cared about. That was all I knew. So armed with these things and this desperation and this fear, I came here. I felt stuck. At the same time, the same curiosity stepped in. We got into the class, right? Started intro bio. Started talking about, evolution and things like that, and plant cells and animal cells. The curiosity came up again. I started asking questions and questions and questions. I would wait outside the class just asking professors questions. Again, not because I wanted to pass the class, but because I just thought about these things, even outside of class. And then that made me go reading. I would spend a lot of time just studying, looking up stuff. Next thing you know, I want to be in a lab. Last thing you know, I get an opportunity to be in some labs, to volunteer in some labs for free. But I just got excited that I'm able to see some of these things that I'll be reading about under the lens of a microscope. And then, one thing leads to another. All right. Next slide, please. This is kind of like a little snapshot of what I'm talking about. In high school is where I developed the interest in life sciences without knowing what it was called. I didn't know what a cell was. I didn't know what all these things was. It was just curiosity about how things work. That was where it all started for me in high school. This is me in college, I think freshman year in biolab or something. So anyway, when I got here, I eventually went to Columbia University, which was, we had state-of-the-art labs and I feel like I was in the place right now where I can actually start, doing things and seeing things and asking questions. There was a lot of resources. So I majored in neuroscience and behavior. How did I go from agriculture to neuroscience and behavior? There's no correlation. Well, like I said before in the beginning, ideas don't come out fully formed. That's why sometimes people might say, I want to do this for my life, at high school level. And then when they get to college level, they start to, the more you learn, the more you refine your ideas. The more you learn, the more you refine your thoughts. Because now you're making you get more information to help you refine your decision. So the more I took classes in Columbia, the more I took classes in college, I just started to see my interests kind of travel from one node to the next. And college would do that to you. That's the good thing about it. It allows you to, it's called a university, right? It's a universe of studies. You get to do your electives. You learn different things from different classes. And then you see what actually strikes your interest. And you follow it. And neuroscience kind of spoke to me as we, focused on brain science. And then, I think around junior year or so, was it even junior year or sophomore year, that was when I declared the major. And eventually I graduated from undergrad. While in undergrad, I did a lot of internships. I had to seek out just opportunities anywhere I can find them to spend time in the lab. I found a few and I made use of those opportunities. After undergrad, I spent some time working because I had to get some money. I had to get a little something, right? I worked for about 2 years in a lab at Columbia University. And then I decided to go to do a master's in biotech. Biotechnology at Johns Hopkins in Maryland. So the work that I did in Columbia now started to show me that so I did neuroscience in undergrad, and then in the lab what I was doing there was testing mouse models of neurological disorders. You could test autism on mouse. It blew my mind. I didn't know that was a thing. We can have an autistic mice and stuff like that. So we do some gene knock-ins and gene knockouts, right? You give them all these disease phenotypes, and then we start testing them, their behaviors. And then the goal was to come up with, some kind of therapeutic drug ideas we'll call it to rescue certain lost capabilities, intellectual disability, all that stuff. So from there, I went to study molecular targets and drug discovery. How do we actually make drugs? Where do they come from? I've been hearing about drugs and medications all my life. My grandma took a lot, different medications. So how do we make that? Again, to me, it was curiosity after curiosity, interest after interest. That is how I got there. And the good thing that happened was right when I was doing this program, again, because I had to make some money, I was working at the same time. I was working full-time, and I was taking this program part-time. So it lasted a little longer, like 2 years. It turns out that while I was in the program, what I was studying correlated with what I was doing at my job. So in school or in class, I would learn about high throughput screening. I will learn about cancer biology. I will learn about cell and molecular biology. And then in the lab at work, I was doing IO, which is immuno-oncology research. I was using high throughput screening for my work. And of course, I had to understand cell and molecular biology. They were in tandem. Remember when I told you when I was in high school, in class, I was studying agricultural science. That was the name of the class. And at home, I was doing agricultural. And then class and school were kind of giving me this robust experience. The same thing happened here. While I was working at NextCure, an immuno-oncology biopharmaceutical company, and doing this program at the same time. Now, at this point, it started to make sense. It's not agriculture, it's not plants, it's not animals, it's none of those things. It's drug discovery. That was finally where the whole thing started to come together. But the truth is, anything you see that seems so finally, realized and finally fleshed out, it's a process. It started from somewhere, just like plants, just like seeds in the soil. It starts small and eventually grows into something magnificent that looks nothing like the original seed. That's always the case. And I'm saying this to encourage you. If it doesn't make sense now, it's okay. If it doesn't sound fancy, it's okay. If it doesn't all come together right now, it's all right. You have the time to make it work. You will get more information that will inform these decisions that will help you make the right ones. Next slide, please. So at Regeneron, what do? I'm going to try to keep it as comprehensive as possible. Regeneron is a company that makes medications. We make drugs for diseases, all kinds of diseases, right? Metabolic diseases like obesity, cancer, muscle atrophy. How many of you have heard about Ozempic? Okay. And Wegovy and things like that. GLP-1 RAs, yep. Those are medications for obesity. My therapeutic focus area is obesity and muscle atrophy, which is how do we help people, battle diabetes successfully? How do we help them lose weight and retain their muscles at the same time? I mean, it's it gets a little complicated down the line. But then some of the therapeutic focus areas is also cancer. So I kind of juggle those both. So at Regeneron, we go from the beginning to the end of the process. That's the thing about the multinational company like that. We start from an idea, a disease that has been nominated, and then some intelligent people start to discuss, well, what actually causes this disease in a person? What broke? What went wrong? And then from identifying what went wrong, what can we do to fix it? Do we need something to bind to something? Do we need to block something? Do we need to replace something? What are the therapeutic modalities that we can go for? And when we answer those questions, the first thing we start doing is to generate that thing, which is the medication, the actual drug, the therapeutic, that would do that, perform that therapeutic role. And when we do it, just like everything else, we start from a large library, meaning we generate them in the millions or in the hundreds of thousands. Why? Because we don't know the lucky one. We don't know the one that's going to hit or the one that's going to be the one that actually works. So we have to go through what we call a screening process. So we start with antibody candidates that are generated from mice. So from this large library of antibody candidates, we want to start testing which ones bind, which ones block, which ones inhibit, which ones activate. Essentially, which one does the work that we need it to do. So the next step is we start to test them with different assays. We have these antibodies. They're in the thousands. We start to test them. So this is what an antibody looks like. Has anyone heard about antibodies? Familiar with them? Yeah. So we know that antibodies have what we call the antigen binding site. They have to bind to an antigen. Okay? The same thing with your COVID vaccines. When you got your vaccines, the goal is to do what? The goal of the vaccine is to do what?
Prevent you from getting it.
Say that again, please.
Prevent you from getting it.
To prevent you from getting it. How does it do that? Okay. So the idea is you get injected with a little piece, a harmless piece of the COVID, a harmless piece. And then your body sees it and goes, whoa, what is that? It makes a lot of these antibodies to bind to it. Okay? So that when you eventually get the real COVID, the real virus, these are already present to bind up that virus and clear it out of your system. That is how it works. But then in biotechnology and pharmaceutical endeavors, we now use these for several different purposes. Because it binds, it becomes a tool. Because in your body, all you have is a bunch of proteins running around, right? They either bind to each other, they interact with each other to either cause disease or to make you function properly. So we now use antibodies like this for those different functions. So when we have all of these antibodies, we start testing them in different assays. The first is the direct ELISA. The question is, does this antibody that we just generated, does it actually bind to what we want it to bind to? It's a yes or no question. If it binds, we keep it. If it doesn't bind, we discard it. It's called a selection process. So we would have a membrane, it could be a cell, it could be anything, would have the protein or the target of interest, and then we have the antibody bind to them or not bind to them, and then we have a detection antibody that will pick up on this one if it bound to them. As simple as that. So the ones that light up like this are the ones that are still bound, and we select those. Of course, we still do more tests on them. A sandwich ELISA, okay.
The most delicious ELISA.
The ideal one, the lunch ELISA. So this one, you coat it on the membrane. Right now, you see the antibody facing upwards. We come in with this antibody to test whether this antibody actually binds to the protein or target of interest. And then another one, the last one, is the cells. We have cells that we generate, and these cells will be expressing whatever protein it is that causes a disease. And then we use these antibodies to bind that target on the cells and then we find a way to detect whether they're bound or not. Okay. So these are some binding assays we use. We also use what we call flow cytometry. So after we do all this binding, we incubate these cells with the antibodies, then we run them through what we call flow cytometry, so that the detection on these antibodies, on these detect antibodies, we can be able to tell, which ones get the detection, which ones did not get the detection, and we sort them accordingly. Okay, that's a lot of jargon. But then the idea is that when we do that selection, as you can see this cone kind of like taper out, we eventually now end up with a few. From the thousands, we end up with 1 or 2. And these 1 or 2 are the ones that pass all of the screening processes. These are the ones that bind. These are the ones that either activate or inhibit. They're the ones that do the work we expect them to do. And then we now create them into different modalities depending on what we need. For instance, if we need something to get into the brain, we have to make it very small, like this, so it can cross the blood-brain barrier. Sorry. It's okay. It's probably the cable. Okay.
It's like a millimeter. Come on now. Anyone ever heard of the blood-brain barrier? It's a great thing. Hey!
Okay. Yeah. So this is much bigger and this is much smaller. We make them into different formats for the purposes. So the next slide. Yes. So the next slide just focuses on some of the instruments we use for these. All right. This is the Z5 flow cytometer. So the method that I mentioned, where we have all the cells labeled and then we pass them through a fluidics to see if we can actually identify what binds and what doesn't bind. We use the flow cytometer for that. These are the cell-based assays, which is the assays that we run with cells. And then we have the assays that we run with proteins as well. And this one is the Opera Phoenix. Sometimes when we bind targets on a cell, we want to take a picture of it instead, right? Because we want to see what it looks like in the body. We want to see the physiological image of what that might look like. So we use what we call the primary cells. It could be muscle cells. If we're looking at muscle cells, it has to actually be muscle cells, right? So we lay them on a plate, we bind them with the antibody, and we take a picture of it. And we use this Opera Phoenix. It's some kind of like a giant microscope. The data looks very beautiful when you look at it. This is an MSD, right? So it's also for cells. You have your cells on a plate, you have your antibodies in there, and then you run it through this machine, and then it gives you the readout of, we call it the signals. If the signal is high, you have a binding. If the signal is low, you don't have binding. So this pretty helps for that. And then for protein-based, like ELISA, we use this one called the Spectrum Max. Right, so you coat your protein on a plate like the ELISA we talked about. You have your protein on a plate, and then you have your antibodies binding or not binding. Either way, it uses what we call the chemiluminescence to give you a readout of the binding. These are some of the instruments we use at work, especially at my team. There's so many other teams. Of course, when we select these antibodies, we have to pass them off to the next, we call it a train, to the next stop on the train, people who are going to now take this antibody, this 1 or 2, make them into what we call the purified antibody, and then we now take them to in vivo testing, right? We start testing them on little mouse and things like that to see how they respond, and then eventually they get to the clinic for clinical trials. And then, with FDA approval and all of that, it can make it to actual patients and help save their lives. All right. Okay. Now, we're going to talk about a science article. A science article that was done, I think, the research that was done recently. I think this article came out in 2025, late last year.
I think you guys all have that actually. When you flip your pages over.
Oh, it's on the page? Great. Yeah, okay, good. So how many have heard about HPV before? Okay, what is HPV? What do you know it to be? Just give me whatever your thoughts are. HPV? Well, the V stands for virus, so it is a virus for sure. It's called human papillomavirus, right? And it infects the cervix. Okay? It infects cervix and causes cervical cancer. Cervical cancer is a big deal. It's actually very painful to treat. You can imagine where it's located, right? Imagine if you have to do surgeries and chemotherapy and all of that. And there's like over 660,000 cervical cancer cases every year in the US. So a lot of women are suffering from it. Now, have you ever heard of HPV vaccine? Right? Young ladies are advised to get it as early as possible to avoid chances of getting the virus somewhere down the line. Now, there are HPV different strains. We have HPV 16, HPV 18. Let's see how I'm doing on time, if you don't mind. These are different strains of the HPV, just like you have your different coronavirus, COVID strains. Now, when they say get a pap smear, pap is short for papillomavirus. A pap smear is essentially a test that shows if a person or a patient has cancerous cells or tumors from their cervix. If it shows positive for cancerous cells, then there's a problem. Now, the vaccines for HPV is supposed to do what your COVID vaccine does, which is what? Like you rightly mentioned, to help you not to get the COVID. So vaccines are usually preventative. You take it before you get whatever it is, the disease is, right? You take it ahead of time. But this study was groundbreaking because it now doesn't just help you prevent getting it. It actually helps treat you if you already got it. It's now called therapeutic vaccine. A vaccine that is therapeutic. A vaccine that has therapeutic effects. Meaning it's almost like a medication at this point. So it was a big deal. Mice that had HPV were vaccinated with this new vaccine. It's a nasal spray vaccine, so they kind of spray it through their nostrils and they inhale it. And you have mucosal surface right around the nostril and also mucosal surface right around the cervix. So they noticed that the mice that got the vaccine, the cervical cancer, the cervical tumor was shrinking after they got the vaccination. And that is mind-blowing because that's not how it works. The HPV is a virus. This is what a virus looks like. And when it infects, I'm going to have to switch back and forth between slides. Can you take me to the next slide, please? All right. This is what the HPV looks like. Again, every virus almost takes the same shape. And what do viruses have inside of them? They only have one material. What do they have inside? They have a piece of DNA. Sometimes it's RNA. Virus is just a casing with DNA inside. That's all it is. So how does it cause all these problems? How does it cause all these diseases? The COVID, the rest of them, HIV, all these viruses. Well, they infect the cells, open up their viral casing, empty their DNA that they carry into the cell. That's it. So what? A DNA inside of a cell, what does that, who cares? What does that do? Well, the problem is the genes in those DNAs. The genes in those DNAs make proteins that are harmful to the cell. In the case of the human papillomavirus, there are several genes called the E1, the E2, the E6, the E7. It's just like generic names, right? The E7 gene on that DNA makes a protein called E7. Now, this E7 disrupts an oncogenic signaling. Let me explain what that means. RB and E2F, when they bind each other, they stop cancer from happening. Okay? RB, retinoblastoma, and E2F, when they bind together, they make sure that the cell does not proliferate if anything is wrong with the DNA. They control cell growth. Meanwhile, what cancer is uncontrolled cell growth. Cells just keep growing and growing and growing and growing and growing non-stop. That is cancer. And because it grows non-stop, it starts to compete with healthy cells for nutrients. And because there are a lot, of course, they're going to get more of the nutrients and so on and so forth. Now, what this E7 does is to bind this RB and dissociate E2F. So, as a complex, their function of stopping the cells from growing uncontrollably gets lost. Now, the RB is inactive. Now the cells are going crazy. That is how the cancer happens. Now you can see how a simple little gene from one little virus breaks a major mechanism and turns the cells loose to uncontrollable growth. That is how the HPV causes cancer. And then of course, another gene here, E6, binds another P53, which also does the same function as this, degrades it, and then cervical cancer happens. That's how it happens. You can go back in the previous slide. Thank you. So, the vaccine targets HPV E7. This vaccine that we said that stops cancer, the vaccine itself, what it now does is the same thing the virus does. The vaccine has a little piece of E7. It has a little piece of E7, a harmless piece of E7. It puts it in a spray, sprays it into the nose, and you breathe it in. How does that shrink tumor? Well, when that happens, your body now sees that E7 as foreign, meaning it comes from outside the cell, and it now makes antibodies for it. Or like, let me take that back. It sees this E7, and then it trains your immune cells to go target any cells that show this E7 on their surface. I'll say it again. You get the E7, a little piece of the E7 injected or inhaled through the spray. Your immune cells recognize this E7, and then they galvanize and drive down to wherever they see this E7, and they kill the cells that are showing this E7. And they go straight to the cervix and start killing these cancer cells that are showing this E7. That is how it works. It was an unintended outcome, but it's great. Okay? So most HPV vaccines prevent infection. This one is designed to treat people who already have precancerous cells and lesions. Next slide, please. Thank you. So that's exactly how it works. And by so doing, chances are this is going to help. There's hundreds of thousands of women who go through this cervical cancer, worldwide. So a preventative HPV vaccine stops most HPV but doesn't treat them. Now, you can see that with this, we're going to save a lot of people the pain of the surgery, the pain of the radiation therapy, the pain of the chemotherapy, just by taking a nasal spray. That is mind-blowing. Do you agree?
Mm-hmm.
Yeah.
Can that be applied to other types of cancer, that concept?
Can that be applied to other types of cancer?
The concept
The concept,
of shrinking the tumors.
yes, it is possible. It is actually possible. Yes. You have to do all these studies to see if it actually works, but it is 100% possible. Yeah. So a successful therapeutic vaccine could add a non-surgical treatment option for patients. So this picture here shows you how vaccines are delivered, right? You have a DNA vaccine, mRNA vaccine, peptide vaccine, and all of that. Who took a COVID vaccine, by the way? Do you know whether you took the DNA or the RNA?
I did RNA.
You did RNA, right? I think RNA was Johnson & Johnson or something?
No,
And then-
Moderna or
Moderna was Johnson & Johnson.
the other one.
Yeah. Okay. All right. So
You got one from every company.
So the vaccine, like I said, your vaccine is essentially a harmless part of that disease or whatever, that's get packaged and then either injected. In this case, it's inhaled. So this vaccine, that E7, is packaged in what we call the LNP, right? And LNP is essentially just something that makes it more, take up more surface area. Okay, it's a gel. It doesn't cause anything by itself. It just it allows that vaccine to spend more time in the area for your immune cells to catch on to it. Okay, and then it gets nasally injected, and then it does all of this other stuff that we just talked about. So my question is, what's the next step? If so far the work is that they were able to package this vaccine, nasally inhale it, spray it into a patient, and then they notice that, again, I said this happened in mice, and then they notice that it shrinks cervical tumor in mice. If you were the scientist doing this study, what would be your next step? There are no right or wrong answers. Yes. Clinical trials for humans. Give them a round of applause. I like that. I like that. Any other answer? Yeah.
Trials on larger animals.
Trials on larger animals. A round of applause for that. I like that. I like that. Trials on larger animals. Any other answer? Any other thoughts? But feel free. Just let me know what you think. Everybody's pointing at you, bro. You got something to say? No? You're putting you on the spot. Okay. Both answers are eventually correct. Those are gonna happen. Of course, mice, do you know we share a lot of our DNA with mice as tiny as they are? Some say 90%. I think that's absurd. But we share a lot of DNA with mice. We have common ancestors. Different story. But yes, so we call them higher primates, right? Eventually we'd have to go from mice to a higher primate. And then eventually humans in the clinical trial. And in the higher primates, we want to do what we call a tox study. Oh, yes?
So why do you guys is the reason why you guys test on mice because of the DNA? The percentage?
So that's a very brilliant question. There is a reason, yes, there's a reason we use mice in labs for testing. The truth is, yes, we share a lot of DNA with mice. That is true.
Yep.
That's why everybody's excited about this. It was tested on mice, not humans. Why do we care? Nobody's trying to treat mice cervical cancer. But the truth is, if it works on mice, chances are high it will do what? It will work on humans. Why? Because we do share a lot of, if you stretch out mice DNA and human DNA, you're going to see a lot of what we call conserved genetic sequence. They're vertebrate, we're vertebrate. I mean, we have a lot in common. It's just like a different stuff. All right. Now, beyond that, we're able to do that with mice because of the quick turnaround, right, in their gestation period. So we want to be able to run these tests in a very shorter period of time, right? From a P0 to a P9, which is like 9 days after mouse was born, you're able to start running some tests. So if you want to see like a whole lifespan, you can in a very short period of time. I mean, you don't want to run these tests on humans. It's unethical. Okay, right? So then from that stage, right, you can collect quick results and then do your iteration. See how it works on mice A, and then try a different set on mice B. And of course, when you do these things, you collect these mice in groups. So you have your control group, maybe about 5 or 7 of them. You have your treatment group, about the same 5 or 7 of them. And then you have other groups. You're able to do these things with flexibility, even while maintaining like a genetic similarity, right? So that is the reason we start with them. And then it's inexpensive. You can control it, and the data is very relevant as well. Then after that, like I said, we go into higher primates. People are, okay, it's a different story. Some people don't like the idea of testing, drugs in mice. Believe me, work is being done right now to kind of phase that out and maybe use AI to do something much more meaningful. But yes, eventually gets tested for toxicity. So before we get to humans in clinical trial, we have to make sure that this therapeutics is not toxic. Okay? One human is way too many to get any toxic, we don't want to see that. So we do what we call the tox studies with the higher primates before we eventually get approved by the FDA to get into the clinic and start testing humans. We have to make sure that it's not toxic to humans at all. So those answers are correct. So I'll give them another round of applause for that. All right, so with the last few minutes I have, I'm willing to answer any of your questions. I must say, when I came here, I was a little nervous. I don't know. I am still nervous. So don't mind me if I stumble upon my words. I don't do this often. I just spend a lot of time in the lab just working with things. I don't know how to interact with humans. Yes?
Pretty much, these guys have a question, but they're too shy to say it, but I'll say it for them.
Wow. No. They will say it. Whoever has the question.
I don't know who it is, but I'll say it anyways, but they said,
Okay.
why do they test on monkeys instead of mice? Like, you know how it's correlated with like weird.
So when we say higher primates, it is monkeys. Yeah. So if you can go back a few slides, Miss Wendy. A few more slides. Yeah. When we do this assay, okay, the cell-based assays, when I say that I have these cells that are expressing, so if this was the HPV assay, I would have these cells, just a bunch of cells, mammalian cells, right? They're just immortalized. They die and regenerate. They just never die. But what matters about these cells is what I express outside of the cell. So what I would do is I would take the DNA, the same DNA that is found in that virus, engineer it, put it in this cell, so that this cell now expresses that E7 on the outside. Now, this is where your question now comes in. I'm going to have cells expressing human E7, monkey E7, mouse E7. Okay? I'm going to have human cells, mouse cells, and monkey cell at the cellular level. I'm not talking about the animals now. Now, I have these antibodies. I'm going to bind them, and in most cases, more often than not, any antibody that binds to the human E7 will bind to what?
The monkey.
The monkey, almost 100% of the time. And that's the mouse, maybe 70%, 60%, sometimes 99%. it's an old tale, but us and these monkeys, it has to be 100%. I don't know, like the genetics, it's enormous. So whatever mostly works on humans works on monkeys, almost like 100% of the time, unless something, untoward goes wrong. So the answer is yes. For toxicity, they're tested on monkeys. I have never seen that with my eyes. I can't imagine what the labs look like. These huge monkeys, and I don't know how it works. I've never been to that level. Most of my work is in the preclinical, right, where we use the cells and sometimes use the mice. But the monkeys and the stuff, I leave it for the pros. But yes, they do test it on them. Yes.
How are you guys working on making testing more ethical?
How are we working on making testing more ethical? I'm gonna say something that is gonna blow my mind. I don't know about you. And when you go home, you could Google it. There's something called a virtual cell. They're working on it right now. I'm talking about a cell that is all on the computer. Yeah. It's, the idea is that we wouldn't have to use these guys anymore. It would all be tested and simulated virtually. So imagine a cell as dynamic as it is. I'm talking about highly multidimensional. You're able to get like genetic sequences of an antibody and run simulation tests on a computer to see how it will interact with a cell without having a cell. If we're able to achieve that, the only thing we'll do in the lab is after we will use the computer to go through all this process, and then eventually just come into the lab, use one cell or two to see what we saw in the computer. Does it translate in real life? That's it. If that happens, it's going to phase out animal testing. Because then eventually we'll become so confident in the simulation that we don't have to test any animals for it. Again, it's in the development stages right now. It might cost me my job. Yes?
How environmental factors impact the cell over time, the cells over time, and the body over time? Can the computer simulate that?
You're asking the right questions. I don't know.
Like I've had Lyme disease that has changed my body.
Mm-hmm.
You know what I mean?
Mm-hmm. I don't know. I don't know what the capabilities of how far it can go. However, I think with that invention or innovation, there's going to be a lot more challenges to be met to answer these more broader questions. But all I can, you have a question, please go ahead.
You think this could go past like cancer? Maybe it could help with also like prion diseases, more like more permanent issues. Do you think something like that could help as well?
Yes, I think that's an incredible question. Yes, it can go beyond cancer to prion diseases and more. When we look at therapeutic modalities or developing therapeutics in the first place, we're not limiting it to a disease in particular. We're trying to see what works based on how the disease progresses, right? Or based on the, we call it therapeutic targets, based on how the target works. In cancer, for instance, I showed you a protein that if it malfunctions, then you get cancer. That is not the same way that other diseases, right, arise. So you have to understand the mechanism of action of a disease. It informs how you pursue your therapeutic approaches. However, whatever works, right, innovation requires that we try to test it through all the different disease focus areas to see if it's applicable. If it is, great. If it isn't, then we try something else. Right? But something like this that proved successful, believe me, is going to be tested on several other disease focus areas to see if it translates. All right, I have 5 minutes. Any other questions? Any questions at all?
Questions about like Emmanuel's journey through science or anything?
Yes.
You are currently working in what you call
Preclinical
pre-clinical stuff,
research, yes.
and you're in a place you never even imagined when you were in high school.
Yes.
Have you thought about like, or visualized next steps in your own personal career or any kind of pathway that you think you might want to be someday in the future?
Thank you very much for that question. Do you know what I think about a lot? Where I came from. I was telling my brother, he's a pastor. So my dad used to be a preacher. He passed away from the COVID, 2021. And my brother took his place. It's mind-blowing. He's too young, but somehow he had to step into the shoes. I was telling him how when I was in high school, I didn't know what was called a cell. I'm talking about maybe like 11th grade or so. I didn't know what, I didn't know there was a school called Columbia University. I didn't know there was a school called Johns Hopkins. We barely have electricity. I don't know, I didn't know much about anything. And now I'm here a couple of years later and all these things are my reality. And my question is how? I ask myself that question all the time. And what it tells me is my future is beyond my, okay? If I couldn't imagine today, 5, 10, 20 years ago, then 20 years from now, what could it possibly be? I don't know. But I know that if I follow my curiosity, I'm not going to miss it. That is what I started with telling you in the beginning. I don't follow trends, I follow my curiosity. I don't follow what people think is cool, what people think is the hot thing, what people think is where to go. I don't care about that. It's too much noise. I can't compete with it. But if I keep asking myself questions that I care about and looking for answers for those questions, I will find myself in the right place in the future. And that's enough for me. I don't care what anybody else does. Okay. But thank you very much. All right.
Let's have everyone head, gather your stuff. If anyone wants to come up and meet Emmanuel, if anyone wants to come up again, feel free to have a conversation. This is a very, very unique opportunity you've got.
What questions do you guys have?
Yes.
You mentioned you work in a lot of different labs, and I was wondering what is your favorite project that you've worked on?
Okay. My favorite project is in the obesity work that we did. I don't know. That was just like personal for me. I don't know why. But, the Ozempic, that is pretty popular. Have you heard of it? Yeah, so we did some of the work on that. And it is very interesting because even in the lab, you could tell how potent and powerful it is. Like it works. So most of the results were exciting for me, that I could see like this has a future, right? And not only that, like developing it in such a way that is even more effective, not just people losing weight, but helping them to fight type 2 diabetes, right? And actually helping them to lose weight while retaining their muscles at the same time seemed really, really like rewarding for me. So when I worked on the obesity target, it was really exciting. Yes, please don't forget your question, okay?
I first or her?
She will go first and then you next.
Okay.
Go ahead.
Hi, I'm Angela.
Nice to meet you, Angela.
It's pretty cool that you're speaking to us at a school right now, but as a scientist, what does your typical day look like?
Okay, I'll keep it short because of time. I come in and I do a lot of, there's a lot of meetings, first of all, because the meetings are where we discuss what we're gonna do, right? The disease targets that have been validated, that have been nominated, and all of the disease background. That is where they're discussed, the plan, the original, the bigger plan. And then after the meetings, I'll start doing what we call experimental design, right? At my desk, at the office, on the computer, I'm designing experiments. How am I gonna do, what experiment is ideal for this project? I'm gonna design that, and then I'm gonna design the actual assay that I'm gonna do in the lab, okay? Then after that, I'm gonna get all the reagents that I need for that experiment. Some are from in-house, some I have to purchase them commercially. And then I have to go to the lab and perform these experiments. And then after performing the experiments, I'm gonna generate a bunch of data. And I'm gonna go back to my office on the computer and start analyzing these data. And then after I analyze this data, I come up with my results and conclusion, okay? And then I'm gonna share it with my team and say, this is what I found from this experiment, this is what I think is going on here. And then we hold another meeting to discuss it. If it's a go, we move on with my ideas. If it's not, I go back and try a different experiment. So that's kind of like the way it works. Of course, yes.
So when you came here from Nigeria, did you go to Colombia right away, or you?
Not right. I applied initially. They didn't take me. They didn't take me right away. So they said I should go try a community college. I did. I went there, I applied for a biology major. They didn't give me that either. They said my background is not very strong for biology. They gave me liberal arts. I took it. And then, you know, I did my thing. I did every class, whether it was an elective, unelective, I was clearing them as much as I can. I did pretty great. And I took every opportunity. I did everything. I was serious with classes. I was meeting with professors. I was, I focused on getting the best grades I can because I did want to go to Columbia. Right.
Really, sorry to interrupt you, Emmanuel.
Yes.
Really emphasize that point, okay? Especially if you're, concerned or not optimistic about where your career or your college hopes are going for any number of reasons, right? Look at where he is now. Look at the universities he's attended, and the origin of his story was not some glamorous, prep school. So this is a very real example of how you can go through all these steps and just never lose hope and never lose sight of what you're trying to do. So
Absolutely, she has a.
How many times did you have to overcome no?
Okay. The no's are endless, as you can imagine. I mean, the whole thing. Even getting to the U.S., I applied for a visa like 5 times. We got refused 5 times. There was a lot of no's. And then, like I said, I came here, I applied to Columbia, and it was like, there's no way. You don't have enough to prove you can survive here. So, I went to the community college, I applied for biology because they said, no, you don't have enough. Like your high school, we can't even you have to evaluate whatever you're bringing to see where it matches. It was a bunch of no's. And then I took what I was given, and then I just started from there. And then, with each great performance, and I kept trying and applying to internships, and then put an application together to Columbia. Now they want to listen to me, right? And then eventually I got in, and I did great, and then Hopkins. But yes, I started in a community college because I didn't get in right away. Yes? Do I see myself doing something now in the future? Maybe yes. Like I told you, I'm interested in doing medical outreach. I have seen what it means for people to just waste away because they don't have access to medicine. It breaks my heart personally, especially after living here and I go home. You realize that a lot of the things we have here is a great privilege. People don't have health care, they don't have health insurance. So I might see myself, advocating for people to get free medication here in the US or elsewhere. I would do that even myself to, galvanize for funds, take a team and go and actually provide free medication for other people. I would do that. I'll do that. Yes. That is a very interesting question because, what's the parallel, right? There's no, livestock in crops versus, high-tech, lab instruments. There's no correlation. But the thing that I think that I got from that is attention to details. Because if you're a farmer, and if you know any farmer, they will tell you, you're not guaranteed harvest at all. Anything can mess up your season, right? It could be termites, it could be weed, it could be anything, right, that comes in and competes with your crops for nutrients in the land. And of course, if your land is not properly tilled, you don't have the right soil, or you don't do proper bush fallowing, it can mess up your season. So we don't have enough resources to waste on maybes. So we have to check out every checkpoint. My grandma will make sure that the bush are properly fallowed, the bush are properly cleared. We come every Saturday to clear out all the weeds, right? We apply fertilizers. You have to pay a lot of attention to detail. The same with your livestock, right? It's a lot to juggle, but you have to follow the details. And I realize that has helped me in the lab as well, because you can imagine how much work we do and how high stakes it is. You cannot miss certain things. It'll be, very expensive or detrimental. So you have to spend a lot of time paying attention to all of the details in your planning. Make sure you have all the right information before you actually start what you're doing. It's transferable, yes.
What's one piece of advice you give to a high schooler who wants to become a scientist but doesn't feel that they have the right background, or but they still want to go into that same field and become a scientist?
The advice that I would have for such student is, you can, period. Okay? You can doesn't mean it doesn't come with challenges, but you can, because when you say you want to do that, you say you want to do science, I believe you want to do it. And because you're sure you want to do it, you're not going to let anything stop you. A lot of things are going to come to try you. The fear of your background, not being good enough, is going to try you, right? Imposter syndrome, not feeling like you can, not feeling like you can compete, not feeling like you're intelligent enough. All of these things are internal because of your background. You feel so low. I remember in Colombia, I'm in class with kids who, prep schools and tutors, you can imagine. Just the idea of that could make me not even want to try. But I'm not worried about them. I'm worried about what I want to do. And I said to myself, this is one of the best schools that will give me that, and I'm going to get in there. Right? So I didn't let these things weigh me down. If anything, I told myself, I am going to do this stuff. I am tough enough for it, and I'm not going to take no. I'm going to put in the hard work to get it done. And that is one thing that helped me. So when you first tell yourself, I can, then the will comes in. Okay?
All right, guys. Well, in the interest of everyone being able to pack up and get where they need to go, including Emmanuel,
okay.
Let's give a round of applause.
Thank you, guys. Thank you so much. Thank you.
If anyone wants to come up in these last few minutes of class and ask him questions one-on-one, or if you guys want to take pictures with him, we've got just enough time before you've got to head out. So at this point, you can take your phones out and relax and come on up if you have any other questions for him, okay? All right, thank you, guys. Thank you so much. That was great. That was a really great presentation.
Thank you, Wendy. I get nervous all the time, so I don't know.
That's so good with
I had to dig through the archives for that.
Somebody in the lab was just like, hey, come here, Emmanuel.
Thank you so much.
Thank you.
Thank you so much for having me.
Public health kids, they're doing non-communicable diseases right now as their project. It's a project-based course.
Oh, wow.
So we're doing cancer, obesity.
Ooh,
right?
Okay. They might enjoy a little bit
who
of.
just presented obesity talked about Ozempic.
Are you serious?
Yes.
Is the student going to be in the
girls who did that presentation? Looking at me.
Wow, what are the odds?
Well, because in public health, we focus more on community versus. So I'm sad that my anatomy and physiology kids couldn't come, because they would follow the technical.
Most of the.
Right, these kids, I don't cover that.
I know.
Focus on the community.
So Wendy did tell me to lay off the jargon with this group, but I couldn't help.
Well, if I had the anatomy kids, they would completely follow what you're
But the interesting thing is, they were able. Maybe they don't have
The
the
basic
experience.
concepts
They were
are
able
totally-
to. They
yeah,
got the point.
absolutely.
They got
Yeah,
the point.
absolutely.
And I could tell from the questions too and the interactions. Yeah.
Thank you
Hey.
for the presentation.
Nice to
I'm
meet
Ryan.
you.
Nice to meet you.
Ryan, Emmanuel.
What class are you guys?
JC here?
JC here.
JC. Nice to meet you. Spencer.
Okay, I just have a question. How long does the cell binding like process take, where you're finding like the 1 or 2 perfect cells? Like how long does that take?
Yeah. So when there are 2 kind of different timelines. One timeline is the assay itself, the experiment, and then another timeline is all of the different assays, because we don't just use one assay at a time. Okay. I mean, we don't just use one assay at all. We use several different assays. So when we get the antibodies, between when we get it and when we send off the leads could be like a month. However, in between those months, we're growing the cells to run the tests on them. We're getting the proteins to run the tests on them. And then we're running the tests. And then we're getting the data, okay, from the experiments. Then we're analyzing the data, right? So what is the signal? Because what we get is like numbers. You have to start making sense of the numbers, right? And then we want to see that our controls work, and the controls don't work, and all of that stuff. So after we do the cell-based assay, then we do the protein-based assay down the line. So by in a month's time or so, we've generated all of this data, and then we send it off to the other guys.
How could artificial intelligence be like incorporated with like testing and like the other stuff? Because isn't like infinite outcomes with how the human body could like react to different stuff?
That is the challenge with AI. But AI is learned, meaning whatever platform you want to use, it doesn't matter. You have to teach it though, right? If I want to teach AI how to select a black and white photo compared to a colored photo, I have to teach it by showing it, feeding it a lot of black and white photos and color, and then it starts to be able to make the distinction. In the case of the antibody testing, we have to teach the AI and say, this is the sequence of antibody we're looking for. If we get this in fact, these are all the sequences that exist. Here you go. How about you tell me the sequence that will match this protein? And then when it tells me that sequence, now it has to create that sequence and give it to me. So the bottom line is we have to teach the AI through what we call machine learning. Then the AI will learn how to identify what we're looking for and then start spitting it out for us. So it's kind of like, and it's a good thing. You're welcome. You're welcome.
Do you have any plans for once the idea becomes more mainstream?
What idea?
Like what you said about the vaccines. If you have any like ideas like that, do you have a plan for once it becomes more more wide for the public?
So you mean the vaccine for the
HPV.
For the HPV
or something similar?
Okay. So what I would think, because like I said, I'm someone who cares about making medication free and available for people who can't afford it. I know a lot of these companies spend so much money developing these things. So sometimes they want to recoup on their on their investment. But my idea would be, how do we make this available to more people who need it? Not everybody can afford it, but if there's a way to make it available, affordable to people who need it, that would be ideal. I will ask the government to subsidize the cost. In other words, pay the company for their investments, bring the cost down, and make it available for people. And then I'll go probably seek out some organizations that can say, you know what, we're gonna foot the bill, right? We'll eat the costs and we'll make it available to as many people, because it doesn't discriminate. Like cervical cancer, a poor person, someone economically challenged can get it. So you want to be able to make sure that they still get the medication. That is what I'll be interested in.
Be in a picture?
Right. You're welcome.
Anyone?
Yes.
All right. You guys were down here.
Thank you for being
Emmanuel, nice to meet you. Addie.
It's very lovely to hear about everything.
Thank you so much. She was still talking. Yeah, I'm gonna get to you if you don't mind.
Like inspirational to hear that even if you don't know what you're doing at first, you can get somewhere eventually. It's very interesting. So thank you very much.
I'm glad to hear that. So what grade are you now? 10th grade. And you still thinking about what you want to do and stuff.
Yeah, I have an idea, but still figuring it out.
That's true, of course. It's absolutely normal. Okay, it's totally normal. It's gonna work out. You don't have to overthink it. Okay, just focus on your classes right now. Do well in the classes. Everything is gonna work itself out.
Thank you very much.
You're welcome. Yes. You said you have a question?
Oh yeah, I was wondering what your GPA was in high school.
Oh my God, bro.
Because you went to a really good college, so I'm just wondering. What
I see. Yeah.
What was it?
It was around a 3.6.
3.6. Okay, that's not bad.
Nah, it's not over the top, but I think what got me in was like they called me a holistic student, like well-rounded, and that meant that the GPA was there. But then I also had spent a lot of time outside of class pursuing my interest. So I had a lot to write about in the essay. All right. And then when we had an interview, I had a lot to talk about. So to them, it was like, wow, if you were doing that and taking classes, we can understand why your GPA is not a 4 point or whatever. But it means you're really curious. It means you're really interested in this stuff. And they gave me a shot.
Did you take in a lot of volunteer activities in high school?
Yes, not a lot because I didn't get a chance to do a lot, but I spent a lot of time in the farm, and I wrote about it. It's almost as if like I found a way to tether it to my academic work. Okay, because volunteering simply means I'm pursuing this interest, and either I see it as this is something that will help other people, or this is something that will help me understand what I'm doing in school. So it could be a volunteer group, it could be an interest that you just have for something you're curious about, which is what I did, and it was able to tie these things together for me. All right, thank you so much. You're welcome.
At the current rate that the trial's going with the mice and stuff, do you have like an estimate of when it's going to be good for human use and human testing?
That's a good question. So for trials, you remember when the COVID vaccine came out and they were like, oh, this vaccine came out too quick. Vaccines usually take 10 years. How do we have this already? It takes a while when you have different kind of vaccines. If I have a vaccine that is protein, I'm not going to confuse you. If I have a vaccine that is protein and I have to grow it out from cells, chances are the cells might get contaminated and I have to grow a lot of cells. But now we are able to make vaccines from DNA, which is very stable and it doesn't contaminate. So I think because we're able to make vaccines quicker, it's going to take quicker to get that whole thing to the clinic. The only thing that will make it take about 5 years or more is all of these testings. Testing it on mice, testing it on monkeys, and all of this testing and getting FDA approval. All of this might take about 5 years, and then the clinical trial itself is going to take even much longer. Because when we say clinical trial, we're talking about testing people. First, you have to announce, hey, we have this drug and we want to test it on people. Who wants to be tested on? Some people show up, some people don't show up. And then, some people come, some people don't come. All these little variables make it take long. But to get to clinic for clinical trial might be 5 years or so.
Got it.
Yeah.
Thank you.
You're welcome. Oh. Nice.
Wait, do you play basketball?
No, I don't. Yeah, sucks.
So I was asking, wait, I forgot my question already.
My goodness, I'm so sorry. Take your time. Take your time.
Yeah, I forgot it. Okay, let me ask another one. Anyway, so on the human cells, do people just volunteer to get their cells? I don't know, how do you guys get the cells?
So the cells that we test are immortalized cells. They're commercial. They're commercial. We buy them.
You can buy cells?
Yes.
That's cool. Okay, I didn't know that.
You can buy cells. So they are mammalian cells, so they're human cells. Whether they're cancer cells or non-cancer cells, kidney cells, liver cells, whatever cells they are, they came from people. Either those people sold it or volunteered it. Have you heard of Harriet something?
Sure, I think so. Yeah.
Yeah, the lady who's like the healer cells. Yeah, so in her case, it was taken without proper consent. But other people, they either sell it or something like that. They volunteer it, and then once we obtain these cells, we now immortalize them, meaning we grow them so much that they can just stay alive like indefinitely.
Wait, how do you do that?
Well, they're genetically modified, right? So they're no longer your primary cells from your tissues.
Also, you can't do that to humans.
No. Whoa. I see what you're thinking. Like, okay. You're thinking ahead of. Got it. No, you can't.
You can't do that?
No, you can't. You can't do it to your entire because your body has like billions of different cells. Right? You have your neurons, you have your muscle cells, healer cells. It's like a lot of cells. So, but this one's just like one cell line. We immortalize it and then we use it for research purposes.
Oh, okay. And then my other question was, like, when you guys make the vaccines, do you like also consider that people might have had like previous diseases and had vaccines for that might like conflict? Or
I hope she's still able to ask her. Oh, she already talked. Okay. Sorry, go ahead.
Like, if they have, like, do you consider that people might have like other vaccines in their body that might conflict, or like other diseases, or?
So if I took a certain vaccine, they don't conflict with other vaccines. For instance, my baby is like 10 months, right? I take her to get her shots.
She's so adorable.
Thank you so much. And I take her to get her shots. The only reason they give her 2 shots at once and not give her 5 or 10 is just because it's too much to stick needles.
Oh yeah.
But if it's possible, we'll give it all at once.
They can't mix it or something?
Yeah, that's the no. They can't. I asked them the same question, like, yo, can't you just mix it and spare her? She at some point she got so familiar with the nurses and the process. When she sees them coming, she starts crying.
Oh, she's like,
I
you're
used to
the
break
one
my
who heard
heart.
me last
Exactly. That's a very good memory. I know you with the blue apron. But no, they don't conflict.
Oh, okay.
So a person can take a vaccine and take another one is to be fine.
Oh, okay. I have another question.
Okay.
If I do biotech,
I'm so sorry.
Oh wait, it's similar to your question. The biotech and nursing, like, are they connected? If I do like biotech and decide I'm gonna do nursing later?
If you do biotech and nursing later,
or like are they connected? If I can do it as like a minor and then do like nursing as a major or something along those lines, like.
I don't know that you can minor in biotech.
Oh, okay.
Yeah, I don't know a school that will provide biotech as a minor.
So about primary nurses,
Oh, I think nursing is incredible. You can do nursing and biotech.
Oh, okay.
But maybe not at the same time.
Oh, okay.
Okay, because they're both like demanding and tasking.
Yeah.
But you can certainly do both. You can do nursing, your bachelor's, and then do a master's, do biotech.
Okay, that's smart. Okay.
You get the best of both worlds.
I see. Okay, nice to meet you. I'm Solange.
Isabella. I'm Emmanuel. Nice to meet you. Absolutely. Sure.
I'll just write it on my notes app because I have nothing else right now.
So my email is just my first and last name at yahoo.com.
Okay.
Yeah. Emmanuel Ozuruonye at yahoo.com. I think it's on the yeah, it's right here.
Yahoo.
At Yahoo.
I would have it. Okay, thank you so much.
Okay, you're welcome.
Have a good day.
You too.
I'm not going.